Hi everyone, good morning. These are going to be the slides on microbial transformations from the professor. Alright, so the first one that has the icon is enantiomers and optically active. These are terms from organic chem. Enantiomers are compounds that are mirror images, and then optically active compounds are going to rotate the plane of polarized light. They're going to rotate the plane of polarization of plane polarized light. Why are we using bioconversion? This is better than chemical synthesis because we have substrate specificity. We only make one reaction in these processes. There's also site specificity. Sorry about that. Only one functional group is effective, even if several are present. Stereospecificity. If a racemic mixture is used, then you only get one enantiomer. Only one of them is converted. Reaction conditions. The conditions for enzymes are mild, so they do not cause destruction of the substrate. Okay, so types of bioconversion. We have redox, hydrolysis, reductions, condensation, and isomerization. An example would be tryptophan to serotonin. Biotransformants include spores, cultures, resting cells, enzymes, or immobilized cells and enzymes on a matrix. Let's talk about the processes themselves now. Microbial conversion. This is going to be when you grow culture in a suitable medium for 6 to 24 hours, and then you add a concentrated substrate without allowing them to grow. Solvents and emulsifiers. These are going to be steroids that can be solubilized with low toxicity, ethanol and acetone. That's what you're going to be using for that. An example would be tween emulsifiers. Then back to the microbial conversion, you add the large inoculum to the concentrated substrate. This costs a lot of money, and you're going to do this immediately without allowing the inoculum to grow. Let's talk about lipophilic materials, such as in the polyphase system. You're going to be using an aqueous phase that has the cell and the enzyme, and then you're going to overlay the aqueous phase in water, immiscible fluid that contains the substrate, and then the substrate will pass through the aqueous phase and is converted. Then the product passes back into the solvent. So you create this like convection current in this polyphase system. The time for biotransformation depends on three things: the type of reaction, the substrate concentration, and the organism used. Hours, you would have oxidation, dehydration, and hydrolysis by bacteria, and then conversion with yeast or fungi would take days instead. Okay, let's talk about recovery. We have extracellular products, so they're going to be dissolved or suspended in the culture. You do cell removal by fungal filtration, or you could also leave the bacteria or yeast in the solution. Cells are washed repeatedly with organic solvent to remove the product. A significant amount may be absorbed by the cells. That's just something to keep in mind. Product is recovered by precipitation by calcium, absorption onto ion exchange resin, extract with solvents, or distillation from the medium if it's volatile. If the product is volatile, you just distill it. Okay, now to some products. Steroids are going to be natural occurring steroids that have hormone properties, like corticoids and sex steroids. They have the same basic structure. A hormone is going to be a product in animals that circulates in body fluids that affect cells that are far away. So this affects far away cells. Let's talk about some natural steroids. We have the corticosteroids. These are going to be the glucocorticosteroid. This is for stress, increased metabolism, and blood pressure. We also have mineralocorticoid. This is for the electrolytes. We also have sex steroids. We have androgens. These are the male sex hormones. We also have estrogen, which is going to be the spermatogenesis, fertility. It's also responsible for female characteristics, and it's a weak minor hormone. We also have progesterone. This is for birth control and hormone therapy. Let's look at the steroid synthesis of diosgenin. This is going to be from the wild yam species, the saponins in the wild yam. I should write that down. And you're going to be producing it by chemical or enzyme hydrolysis. Okay, so now it's converted into Reichstein component S via chemical steps. Next, we are going to go and take the Reichstein's component S. We're going to do 11 beta hydroxylation with the Cuvillaria lunata. This is a fungal plant pathogen. Cuvillaria lunata will convert it into cortisol, hydrocortisone to be exact. Then your cortisol will go through dehydrogenation by Corynebacterium simplex, so another organism, and that makes it into prednisolone. Prednisolone will then go over chemical oxidation to go back to cortisone, and then cortisone could then be dehydrogenated by Corynebacterium simplex, and then we're back at prednisone. So we get to our prednisone here. Okay, use of steroid hormones. We have cortisol. You could use it for eczema, skin rashes, inflammation. Prednisone, we use that for asthma. And prednisolone, we use that for allergy, autoimmune disease, and cancer. Fludrocortisone, that's for adrenal insufficiency or Addison's disease. Testosterone, that's for low testosterone prostate cancer. Estrol, that's for contraception, hormone replacement, and feminizing therapy. It's also the same thing as estradiol. Progesterone, that's going to be birth control, hormone therapy, and then progestins are synthetic. Those are going to be for hormone replacement and therapy. Let's talk about ascorbic acid, also known as vitamin C. This is found in citrus fruits, berries, and vegetables. You need this to repair the tissues. You also need it to form collagen and neurotransmitters. This is going to be used to treat scurvy. This was common bleeding of mucous membranes in the early 19th century. You also get anemia from this. China produces 70% of ascorbic acid of the world's supply. Let's talk about the Reichstein process. The Acetobacter species will oxidize the D-sorbitol to L-sorbose at pH 4.6 at 30 degrees Celsius. However, the new method will be the two-step process. You have Gluconobacter oxidans. This is going to be sorbitol to sorbose. This is a 98% yield. And then step 2, this process will be ketogluconogenium vulgare and Bacillus megaterium. Sorbose will become 2-keto-L-gluonic acid. Sorry about that. The 2-KLG yield is greater than 97%. And then the Bacillus species, this Bacillus megaterium, this will give it enzymes, nucleotides, amino acids, intermediates, vitamins, and sporulation in order to supply the cold culture when it's doing this product. And that's going to be all for bacterial transformations. Please do something nice for someone. Have a good one.